right, so this is now muscle lecture number 3. The goal of this one is going to be go over the anatomy of a skeletal muscle cell, and then also go over what is called a sarcomere, which is one functional muscle contracting unit. It's going to be, unfortunately for you all, a lot of my bad drawing. However, again, I have a picture of it in the outline, plus I have supplemental things added on the canvas shell to also see it as well. I also incorporated a chunk of my cell biology lecture, which is a lot more technical and molecular based. But if you want to look at it, that's also on there as well too. Granted, you don't have to know all those gross details. What I'm going over is what you got to have a working knowledge of. All right, so continue on with the outline. Where we left off in the outline, we were talking about the characters of muscle tissue. Then there's one little section in there that talks about skeletal muscle. There are two groups of collagen parallel elastic components and series elastic components. You can read that as well as I can. It's not a big deal. Now let's go to the skeletal cell microscopic anatomy. These vocabulary words you have to know. Well, first one, nuclei. Not a vocabulary you have to know. It's not anything crazy you don't already know. Nucleus, you know, is one. Nuclei means more than one. We already said that skeletal muscle cells are multinucleated, meaning that they have more than one nucleus. Why? Because it's actually several thousand cells which actually fuse together to make one long cell, the longest being 30 centimeters long. So therefore it's multinucleated. However, so when you think about the muscle cell, think of it like one big long cylinder. That's the muscle cell. Now, in that muscle cell, understand there's going to be lots of stuff in the cytoplasm, meaning protein fibers, and they take up a lot of room. So what do is they take those nuclei that would normally you would think be found kind of floating in the middle of a cell when you picture of it in your head. It can't. It gets squished to the outside because the entire volume of the cell is being mostly occupied by protein fibers. All right, so the first new vocabulary word you have to know is the word called sarcolemma. Sarcolemma is the fancy-smancy word to say the cell membrane of the muscle cell. In that sarcolemma, when we go over the next lecture beyond this, it's going to be important because that's going to be the area for a nerve impulse to go from the end of a nerve, an axonal terminus, into the muscle cell. So it's going to be a major conduit to get the nerve impulse into the cell. So for now, it's the cell membrane of the muscle cell. Every so often, what the muscle cell will do, though, as the sarcolemma runs along the cylinder, every so often the sarcolemma will merge and it will invaginate. It will actually invaginate and go all the way from one side of the tube to the other side of the tube. Well, what's it doing? It's cutting across the tube. Hence why it's called a T-tubule, transverse tubule. That's going to be important more so for the next lecture, not this one. All right, so what I'm saying then, start the bad artwork, I guess. What we're looking at is the muscle cell. And it's going to be like a big cylinder, okay? Like that, all right? This layer up here, that's going to be the sarcolemma. That's the cell membrane, okay? What you would see then are these large, like beans on one side. Those are the nuclei. They're squished to one side. Every so often, like I said, this sarcolemma is going to invaginate and go from one side to the other side. That's a T-tubule. And again, as you progress down, it'll happen again, and again, and again, and again, all the way down. All right, but we're going to focus on one of these. All right, now, moving on then, let's see, your next vocabulary word I gave you is sarcoplasm. Sarcoplasm is the fancy-smancy way to just say the cytoplasm in a muscle cell. So when you hear the word myo, that means of a muscle. Sarco means of a muscle cell. It's Latin root words for all those. So the sarcoplasm is going to be this material here. As I said, it's mostly going to be filled up with strands of protein. Again, go back to my previous lecture when I said think of like coaxial cable. We have one tube and several wires running through it. Same idea. So running through here then, there's going to be all these little protein fibers running all the way through them. Okay? Now, those protein fibers, please make sure you note star bedazzle. Those are going to be called myofibrils. Myofibrils, or each one is a myofibril. It's the length of protein. All right. Now, also found in the sarcoplasm, you got to understand there's going to be two things. In order for the muscle to contract, it has to have a form of energy via ATP. Whenever you start to work out, whenever you start to run, what you got to remember is your hemoglobin hasn't had a chance to go from your lungs to your muscles to give the oxygen that it needed. There's a time gap there. But as you're working out, your muscle's still working. How? It needs energy, right? From a previous lecture, your skeletal muscles, if you remember, can store a complex carbohydrate called glycogen. It's a sugar. Additionally, in your muscles, you have a red pigment called myoglobin. Not hemoglobin, that's in your bloodstream. Myoglobin. Myoglobin you know because when you go shopping for food and you look for red meat, steak or whatever, do you buy the meat that's gray or do you buy the meat that's pinkish? You usually buy the one that's pinkish. I hate to tell you this, but you understand why it's pinkish when you bought it, especially if it's been sitting there. That's a red dye. The red dye gives you the illusion that it's fresher than it actually is. It's still, the due date is still valid, still the same. But if you had a hunk of gray meat next to a piece of pink meat, you're going to gravitate toward the pink meat. I mean, you're not dumb. The butcher's not dumb either. Now understand, the only way for you to actually see this, to be pink, you'd have to be arm in arm with the butcher as he or she is slicing it down, and when they slice it down, it'll be pink. But the myoglobin, once it hits the atmosphere, it oxidizes like that, and it starts to turn gray. So even if you got the meat right off of the butcher as he or she was doing it, put it on the little styrofoam, wrapped it up in plastic, by the time you're walking up to the front, going through checkout, it's already turning gray, even though it's fresh. So it's just a marketing tool to make it pink. But the myoglobin is what makes the red meat red. It's what stores the oxygen temporarily. That's all going to be also found in the sarcoplasm as well too. Now, surrounding these myofibrils, there's going to be a network of tubules. It is going to be it's the smooth endoplasmic reticulum of a muscle cell. Because it's a muscle cell, it's called sarcoplasmic reticulum. Kind of looks like a bunch of spider webs going all over it, all over the myofibrils. However, as it gets closer and closer to a T tubule, all the tubes will merge and make these sacs at the end. These sacs at the end. These sacs at the end are called terminal cisternae. What does the sarcoplasmic reticulum do? It acts like a big sponge. Its job is when calcium ion, Ca2+, is dumped into the sarcomere, then the sarcoplasmic reticulum, like a sponge with calcium pumps, will go suck it all back up again. And when it sucks it back up again, it stores it in the terminal cisternae. Now follow something here. That means that this muscle cell, this muscle unit next to it has the same arrangement, myofibrils. It's also going to have a sarcoplasmic reticulum that's going to end in a terminal cisternae right there. This structure is important. Notice I have a T tubule, a terminal cisternae on either side. That is called a triad. Okay, so with that should have killed pretty much going through everything with muscle cell anatomy. All right, now, next thing we have to understand is we're going to take this area, this myofibril, and we're going to, in your imagination, on a much more molecular level, zoom in and take a look at it. We're now going to explain why skeletal muscle is striated, why it goes light, dark, okay? So, with that, I'm going to get rid of this right now, and we'll move on to a new one. All right, so the myofibrils are made up of smaller proteins called myofilaments. Don't confuse them. Myofibrils are bigger. They're made up of myofilaments. There are three main types of myofilaments. There's going to be thick, thin, and elastic. Thick is not really thick. It's about 15 nanometers in diameter. It's made of a protein called myosin. Big star. Myosin. Myosin, whenever you see them, think of Picture like taking two long golf clubs, like two drivers, and intertwine the handles. So basically you get these intertwining handles, and at the end of it you get the globular heads, the end of the club. Now, those globular heads at the end are important. There's going to be two binding sites for them. There's a binding site for ATP, which is going to activate it. Basically what it does to the myosin head, think about taking like a firing pin of a pistol and pulling it back to activate the energy. Other part of the myosin head is also going to have an area that's going to bind the thin filament, the actin. Okay? So, the next one is called a thin filament. Thin filaments are they're 7 nanometers in diameter, about half the size. They're composed of a protein called actin, A-C-T-I-N. Now, actin's a little different. There's two different types of actin that make this up. One is called F-actin, one is called G-actin. F stands for fibrous actin, G stands for globular actin. Imagine now I take two long protein chains and I make a lazy helix. That would be F-actin. Okay? Now, when you think of a helix, you think of it like the double staircase. Take that double helix and tilt it towards you. What do you have? A hollow cylinder, and it actually makes the outside of the cylinder. Inside of it, you're going to have tucked these large pieces of protein called G-actin. G-actin is what's going to have the binding site for the myosin head. So on these, we got these little notches on each one of these, and those are the binding sites for the myosin head. Now, question, got a problem. Skeletal muscle is supposed to be voluntary. We should be able to contract it when we want to. There has to be a way to regulate this to binding to this. Well, that's our next protein. Covering up the binding sites on G-actin is going to be a protein that covers it up called tropomyosin. Tropomyosin binds the binding site on the G-actin when the muscle is relaxed. Well, that's a problem. Now I can't reach across. How do I reach across? Well, sitting on tropomyosin is a small little protein, and my blue marker's dead. So, sitting on it is going to be another little protein sitting on top of it. That protein is called troponin. Troponin is a little tiny protein, and it has a binding site for calcium, Ca2+. Think where I'm going with this. If the terminal cisternae store calcium, when they're activated, they're going to dump the calcium into the muscle cell. When they dump the calcium into the muscle cell, the calcium will bind to troponin. Troponin in turn will change shape. When it changes shape, denatures, it will make the tropomyosin shift. What used to be covered, the binding site, is now exposed. And now the myosin head can reach across and grab. Okay? Now, whenever the myosin head reaches across and grabs the actin, that is called a cross bridge, because we're bridging between two different proteins. All right, so far so good. Okay, now, with this then, the third element that we have to deal with, as you see in the list, is the myofibril called elastic filaments. They are composed of the protein called titin, T-I-T-I-N. They are only 1 nanometer in diameter, so they're the smallest ones, and barely people ever talk about them, but they're kind of important because they hold the whole thing together, okay? So, with this, I'm now going to go over why the muscles are striated. Why are there dark and light bands, okay? So, do yourself a favor, draw this out with me. It will help you out immensely. All right, so what I'm first going to do is I'm going to put in the thick filaments. That's going to be in black, okay? So I'm going to make those thick filaments and have a little space in between them. Okay? Now, if you notice, I have gaps in between. To my right, to your right actually, where there was a gap, I'm going to put another stack of myosin here and here. Same thing over here and here. Okay? So they're staggered. All right? Now, in between these stacks, there is going to be a protein that zigzags back in between. It's called the Z-disc. Now notice, it's not going to touch the myosin. wherever the stack is, my zigzag will point to it. So you point to it, point to it, point to it, point to it, point to it. Ooh, that was horrible. Let me fix that. Zigzag. All right, so zig, 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 and zag. All right. Now that's tighten. That's the thin filament that's holding. That's the thinnest filament holding together. Now watch. The tighten will actually reach out and grab onto the myosin bands. So what you got to imagine is like these Z discs act like two trees, and the myosin in the middle acts like a hammock suspended between two trees. Okay, so it doesn't make direct contact. Coming off of the Z disc, though, what does make contact are short little strands of actin above and below. Now notice the actin does not come all the way across. That's going to be important a little bit. Now follow. The blacks are myosins, the thick filaments. The red would be titin, the elastic filaments. The green would be the actin, the thin filaments. Okay? Now remember, actin, the green, looks like this. The myosin looks like this. Like thousands of them wrapped around each other. This is just one strand, but just imagine a bunch of them wrapped around each other. Okay, now, when you looked at striated muscle, when you looked at skeletal muscle, it was light, dark, light, dark. All right, so follow. If I look at this, when all the myosins are stacked up on top one another, that's when you saw under a microscope why it looked darker red or a darker pink. So from here to here, where the myosins are stacked up on top one another, that is called the A-band. A-band stands for anisotropic. It's unimportant. It just means that whenever you were to run it through a lens with a polarized lens, the light, when it comes out, would emerge counterclockwise. That's what anisotropic means. Unimportant. Just know it's called the A-band. That's only where the myosins are stacked up, which also means then this one would be the A-band over here. This one would be the A-band over here. Notice the Z-disc doesn't have any myosin. That's whenever you saw the striations and it was lighter. So it went dark, light. That lighter area in between, this is called the I-band, in between. Stands for isotropic, meaning it's going to go clockwise, not counterclockwise through a polarized light. All right, now, in the middle of the A-band, if you notice, there was this little area right here where the actins didn't touch. This is called the H zone. Why is that significant? Because when the muscle goes to contract, it's going to contract like an accordion. It's going to shrink into each other. The myosin isn't going to go anywhere. It's going to be like a tug-of-war match, but now in the tug-of-war match, you have a rope in one hand, a rope in the other hand, and you're going to pull toward the middle successfully. Eventually, this H zone is going to get smaller and smaller and smaller till eventually the left and the right side touch. The contraction stops. This is when we go over it. The next lecture is going to be called all or none, meaning we contract all the way in or you don't even start all the way. Okay? So, to recap, all the blacks, those are myosins, thick filaments stacked up on top of each other. When they're stacked up on top of each other, that's the A-band. Whenever you have this Z-disc in between it, there's no myosin. It looks lighter in color. That is called the I-band. In the middle of the A-band, where the actins come off the Z-disc but don't come all the way across and don't touch, there's a gap. That gap is called the H zone. That determines how much we can contract, like an accordion. Understand that means when the muscle relaxes, the accordion relaxes and the H zone gets reestablished. So it just keeps going in and out. All right. The Z discs are made of titin, as we said. The only thing left that I haven't labeled for you is what's called a sarcomere. A sarcomere means this. If I go from one Z disc to the next Z disc, so a Z disc to a Z disc, that is called one functioning contractile unit, one muscle unit. That is called a sarcomere, which again means one muscle contracting unit. Let me look at my notes real quick to make sure I didn't skip anything because I went rogue on you like I normally do. but I hit it all. All right, so next time, my next lecture will be specifically then, it's going to be a big one. It's going to be on a molecular level, how the muscle contracts. For this one on Canvas, I also have three little video clips to help to go with it, to help explain, and it's also animated, so you don't have to hear me talk and pantomime it. All right? All right, so I'll see you next time.